Welcome to Biology at Priyajit, my dear viewers. Today we learn how RNA polymerase III in eukaryotes initiates transcription step by step. This enzyme is responsible for making small but essential RNAs such as tRNA, 5' rRNA and some snRNAs which are crucial for protein synthesis and RNA processing. So we will explore how polymerase III uses internal and upstream promoters assembles with specific transcription factors like TF3B, TF3C and TF3A and starts transcription precisely at the correct site. By the end of this class, you will understand how eukaryotic cells accurately produce these vital RNAs, ensuring smooth cellular function. Let's dive in and see how polymerase III kicks off transcription inside the nucleoplasm. Initiation of transcription by RNA polymerase III or Pol III. RNA Pol III works in the nucleoplasm of eukaryotic cells. Promoter recognition. Pol III cannot recognize promoters on its own, but it relies on transcription factors which firstly bind to the promoter and then recruit polymerase III to the start site. Types of promoters. Pol III promoters fall in two main categories. Internal promoter that is found inside the gene or downstream of the start point. Examples include 5' rRNA and tRNA genes, and upstream promoters they are located before the start point, which is more conventional, and example include snRNA genes. So internal promoter details: 5' rRNA promoter located between plus 55 to plus 80 in the gene, and this sequence can initiate transcription if placed in other DNA regions. So there are two types of internal promoters. Type 1, that is box A and box C, that is separated by variable sequences and found only in 5' rRNA genes, and type 2 where box A and box B, so found in tRNA genes. So how internal promoters work? TF3C basically binds to the internal sequence where box A and box B for type 2 and box A and TF3A at box C for type 1. Binding of TF3C allows TF3B to bind at the start site, and Pol III is then recruited to the start point. Key roles and factors: TF3A and TF3C assembly factors that position TF3B. They are not needed for actual initiation once TF3B is in proper position. TF3B, the main true initiation factor, it remains at the start site and allow polymerase III to bind and begin the transcription process. Beta prime subunit of TF3B, it helps open the DNA for transcription like the sigma factor in bacteria. Upstream promoters in the type 3, it is found in snRNA genes, consist of three upstream elements like Tata box, PAC or proximal sequence element and oct element. So Tata box ensures the polymerase III specificity. Proximal sequence element and oct elements increase transcription efficiency by helping factors bind cooperatively. TBP or Tata binding protein in TF3B positions Pol III correctly at the start site. So what is the general principle? Regardless of promoter location, whether it is internal or upstream, these factors bind first, forming a pre-initiation complex. Then Pol III does not recognize the promoter directly. It binds next to the factors positioned near the start site. So what is the summary? Use 3FC plus or minus TF3A to position TF3B, then Pol III. Upstream promoter TF3B with TBP binds directly at Tata box and recruits Pol III. Take home point: what is the point? The location of the promoted elements can be different, but transcription always starts where TF3B is positioned, guided by the transcription factors. So as we conclude, remember that transcription by RNA polymerase III in eukaryotes is a finely tuned process from recognition of internal and upstream promoters to the precise assembly of transcription factors like TF3C, TF3B and TF3A, and finally to accurate initiation of tRNA, 5' rRNA and snRNA synthesis Every step is carefully orchestrated. Understanding this process not only strengthens your concepts for undergraduate, postgraduate, or NEET exam, but also helps you appreciate how eukaryotic cells reliably produce their essential RNAs for protein synthesis and how RNA processing occurs. So keep revising, stay continuous, stay curious, and continue exploring the intricate world of molecular biology. Thank you.